I will start in 2 minutes. Okay, so I'll share my slides. This is the final session of this course, NMR Spectroscopy for Structural Biology. So far, in the last 4 classes, we have covered basic principles of NMR, NMR in DNA and RNA, and NMR in proteins. This class will go further into NMR in protein ligand interactions and go into the basics of drug discovery. So we know that protein molecules are highly diverse communication platforms. It is the interaction with protein molecules with other molecules like small ligands or sugars or lipids that result in various functions in the body. Proteins can also interact with DNA, which basically forms the nucleosome, where there is a DNA strand wrapping around proteins which are known as histone proteins. So these are some ligands that proteins can interact with, like the drugs you take, the painkillers like aspirin, ibuprofen, or other medicines like diclofenac. They all interact with proteins in the body, and that is how they have their effect. So protein ligand interactions are very important for any biological function. These another kind of interaction are protein-protein interactions. These regulate cell communication and they are the glue to drive biology. Protein-protein interactions are responsible for receptor activation, signal transduction, DNA replication, and invasion. So if you are touching a hot object, uh, the receptors on your skin are indicating that it is a hot object and you need to remove your hand. This entire process happens due to protein-protein interactions. Even in DNA replication and invasion means the immune response of the body, this all happens due to protein-protein interactions. Now, the basis of protein-protein interactions is shape complementarity. The one protein fits into another protein when there is a complementarity in the shape. So, and after binding, a conformational change happens, which leads to the signal transduction. Now, there can be two models of this shape complementarity. One is a direct fit, where one protein exactly has to fit into the groove of the other protein. It is also called lock and key model. Another model is the induced fit model, where you can see the protein does not directly fit into the groove, but eventually the groove adjusts its shape to properly accommodate the protein. So first it binds weakly, then you have to remember that proteins are dynamic molecules, so they can change their shape and conformation. So this receptor protein, then after the weak binding, the receptor protein changes its conformation uh, and the fit is improved. So this is called the induced fit model. Another effect is the allostatic effect. So this is the receptor protein and this is another protein or the ligand that is responsible for the biological activity. So allostatic effect means that normally the ligand would just bind to the active site and the function would happen. An allosteric effector, what it does, it binds to the allosteric site, which is not the active site. It is another receptor site. But however, this allosteric binding causes a change in the active site conformation. As a result, the original ligand is not able to bind to the active site anymore. So allosteric effect is basically the binding to a site, and the effect of this binding is seen in another site. So in this particular case, it's a negative effect. The binding of the allosteric effector is preventing the binding of the original ligand. Sometimes it can even be a positive effect, that this kind of binding is improving the binding of the original protein ligand. So this is called allosteric effect. Now, can we quantitatively understand protein-protein and protein-ligand interaction? How can we understand that? One is the stoichiometry, that how much of protein is binding with how much of the ligand, whether it's a 1:1 interaction or 1:10 interaction, and so on. Then is the kinetics, that is the rate at which the protein binds to the ligand, which we can call K on, and the rate at which the ligand dissociates from the protein, which we can call K off. Another point is the thermodynamics, the enthalpy, free energy, entropy of the binding. All these have to be favorable in order for a good protein-protein or protein-ligand interaction to happen. There is not a single biophysical method that can offer all types of this information. You may, from a particular experiment, you might get stoichiometry, but you won't get thermodynamics, and so on. So there is no single method that can give all this information. Other informations that are important are magnitude of the affinity, that is the strength of binding of the protein-protein or protein ligand. Reaction rate, which already falls within the kinetics, but another part of reaction rate is the lifetime of the state. That is, what is the protein ligand or protein-protein complex that is forming? How long does it last for? NMR spectroscopy is the method of choice for addressing the protein ligand or protein-protein interactions. And why is that? We will look into that in a few slides, that why NMR spectroscopy is used. Let us look at the basic kinetics of protein binding to a ligand. So P is for the protein, L is for the ligand. It is reversibly binding to form the protein ligand complex PL. One assumption we are taking is that there is a single binding site, that is the binding stoichiometry is 1:1. One molecule of protein binds with one ligand. The dissociation constant is defined as the concentration of the free protein into the concentration of the free ligand divided by the concentration of the protein ligand constant. Kd is simply the free ligand concentration at which 50% of the protein population is bound to the ligand. So if 50% of the protein population is bound to the ligand, then the PL concentration will be equal to the P concentration, so they will cancel out. So then Kd will become the concentration of the free ligand. Kd is basically the dissociation rate. So if the protein ligand concentration is dissociating into P plus L, then that equilibrium constant is given by Kd. The association constant Ka would be the reciprocal of this. That is the rate at which they associate, that is the free ligand and protein form the complex. The unit of this is mole inverse, because you can imagine each of these are in molar. So 1 molar will cancel out. So Kd, the unit is molar. So Ka, the reciprocal, the unit will be molar inverse. As I said, Kd is 1 by Ka. The strength by which a protein interacts with a ligand can vary over various orders of magnitudes. The ultra strong binding is where the Kd is less than nanomolar. The intermediate range of binding is when Kd is in the micromolar range. And the ultra weak binding is when Kd is greater than millimolar. So these are different ranges of binding. It can range from nanomolar to less than nanomolar to even greater than millimolar. Most of the biological phenomena occur with Kd around micromolar, that is in the intermediate range of binding. NMR spectroscopy is capable of providing quantitative information for protein ligand interaction with affinities lower than micromolar. So that is one advantage of NMR spectroscopy. Now, when there are two molecules in solution, they will undergo tumbling or diffusion to come close to each other. And via this tumbling or diffusion, they will occasionally encounter each other. Depending on the precise orientations of the two molecules, this will lead to formation of what is called an encounter complex. Means that when they are hitting each other, some kind of a complex will form. That is, so you can see here, this is the free energy of the unbound protein and the unbound ligand. They are diffusing, and at a point they hit each other. That forms the encounter complex. This is not the final bound complex, so it has higher energy than the bound complex. But here some interactions have started to form between the protein and ligand, so it has lower energy than the free unbound forms. Then slowly from the encounter complex, as the ligand fits more and more into the cavity of the protein, the final bound complex starts forming, which has lower energy than the encounter complex. Now, there are two ways in which this can happen. One is, so here what is represented is the conformational selection. So there are multiple protein conformations. As you know, protein is a, just one second. As you know, protein is a dynamic molecule, so it can go through multiple conformations. The ligand searches through all probable energy states and will bind to the one in which it fits better. So the blue one is the different conformational states in which the protein can exist. They have different free energies. The red graph is for the ligand. So the protein can exist in different conformational states, but the ligand prefers to bind to the protein in this small range. So this is called conformational restriction, because at the conformation where both protein and ligand can coexist, it is less, the conformational range is less than the total possible conformation of the protein. So a conformational restriction is happening. That is, there is loss of degree of freedom of the protein on binding. So you can see here, if the protein can exist in this much area, the ligand is taking up a smaller area. Or there can be conformational extension. So if the protein can exist in all these conformations, the ligand can actually exist in even more conformations. In that case, to accommodate the ligand, the receptor protein extends conformation. So that is also a possibility, that to accommodate the extra conformation of the ligand, the protein conformation is extending itself. That is called conformational extension. You can see it is represented here. The blue protein has a smaller conformational range, whereas the red or ligand has a wider conformational range. Another possibility is a mixed effect, that is, both conformational restriction and extension happens. So if you can imagine, part of the ligand conformation is within the protein conformation. There is kind of an overlap like this. So you can imagine it as conformational restriction within this area, but conformational extension within the rest of the area. So this is a kind of mixed effect. Another possibility is induced fit or conformational shift. That is, protein conformation, free protein is existing here, free ligand is existing here. In order to overlap, bind the ligand, the protein has to change its conformation. That is, there is a shifted conformation of the protein to bind the ligand. That is called conformational shift or induced fit, where the protein can change its conformation to fit better. Now, to come back to the previous question, why do we use NMR to study protein ligand interaction? Most biophysical and structural techniques to study protein ligand interactions fall into two groups. Techniques that can measure the thermodynamics and or the kinetics of the interaction, or techniques that can elucidate the structure of the interaction. NMR can simultaneously do both, which makes it a very good method to study these interactions because one experiment is giving you all the information, be it thermodynamics, kinetics, or structure. So uh, we know that from NOE or NOSI, we can get which is based on dipolar interactions, we can get distance restraints. So the NOE intensity is directly proportional to 1 by r to the power 6, where r is the interproton distance. So from NOE intensity, we can get the distance constraints. And using the distance constraints in an energy optimization algorithm like Tandy algorithm, we can get the minimum energy structure. So from NMR, we can determine the structure, and we can also determine the thermodynamics or kinetics quantitatively. We have not looked into that much, but it can be done. And the structural details we also get are at atomic resolution, since the NMR is due to the nuclei. And for NMR, what is needed? It's not like for others things like cryoelectronic microscopy and things you might need, or for X-ray crystallography you will need good crystals, or for cryo-electron microscopy you will need a very low temperature setup. But for NMR the requirements are much lesser. You need a sample concentration, that is the protein concentration, of 500 micromolar or greater, and the ligand concentration uh, is titrated according to its binding with the protein. The protein ligand complex must have stability up to 24 hours, because sometimes the 2D NMR experiments can be taken up to 24 hours also. So the complex must be stable for that entire time. Temperature range 5 to 40 degrees Celsius is enough, but it is important to remember that higher temperature can affect the exchange rate. And pH of less than 7, which will be good for the protein stability, that is maintained, because if the pH is too high, proton exchange can happen. And it should be ensured that the buffer that is used to maintain the pH less than 7 is not interfering with the protein signal. And in the NMR tube, a volume of 500 microliter is taken. Now, how can NMR be used to describe the protein-ligand interactions? There are certain properties of NMR that are used to describe these interactions. One, we know that in any NMR peak, the important properties are the position of the peak, that is the chemical shift, which can be defined in terms of nu, that is hertz, omega, that is radians per second, or the most commonly used one, that is delta, that is defined in ppm. This is a characteristic of a particular nuclei, and the chemical shift may be different in the free protein and the protein ligand complex. So a change in the chemical shift will indicate that binding is happening. Another is the intensity of the peak, that is the relative concentration and thus the population of the peak. So if the concentration of the peak is reducing, that means suppose this is the peak of the free protein. If the concentration of this peak is reducing, that means the free protein concentration is less, and so more of the protein is getting involved in forming the protein ligand complex. So there the intensity of the peak is important. Another is the line width, that is this lambda, which can be defined in terms of hertz or ppm. And we know that line width, one can be the reasons for the line width. One is the field inhomogeneity, that can be corrected with a good NMR spectrometer. The other is the T2 relaxation time. That has that can change on binding. So the relaxation properties of the nucleus depend on whether it is binding with a ligand or not. And so if that changes, the line width will change. And so that will also be an indicator that binding is happening. And as we know, conformation can change after association. In that case, any of these properties, the chemical shift, the intensity of the peak, or the line width can change. So there are certain methods for measuring the binding when the ligand is smaller than the protein. So if the ligand is a small molecule or peptide of less than 10 amino acids, Binding can be monitored by using either signals from only the ligand or signals from the protein or signals from both. And this can give information about which nuclei are involved in binding. So the ligand is small, and so its relaxation times will be slower. This will result in sharper lines. Line width will be less. Also, they are small molecules, so diffusion will be fast and the NOE will be positive. And since the number of spins are less, since it's a small molecule, the spin diffusion is also negligible. Protein, on the other hand, is a larger molecule generally, and so it will have fast relaxation, which will result in line broadening. And due to it being a large molecule, it will have slow diffusion and negative NOEs. And since the number of spins are large, the spin diffusion will be fast. Ligand protein complex will behave similarly to the protein since it is also a large molecule. And so all these properties that we discussed in the protein will also be present for the ligand protein complex. Now, there are certain experiments which are called ligand detected NMR experiments. One such experiment which we will study is called the saturation transfer difference. Here, we do not label either the ligand or the protein, both are unlabeled. And one requirement of this experiment is the ligand and protein have to be different in size. So for the saturation transfer difference NMR spectroscopy, there has to be a size difference. Another requirement is that signal of ligand should not overlap with the signal of the protein. We need an excess of the ligand for this experiment. Only 30 picomoles of protein is needed, but ligand needs to be much higher. And we talked about size difference. Generally, proteins are always much larger size than ligands. So since the ligand is small enough, there is no spin diffusion in the ligand. So you have the ligand and you have the protein. They reversibly form a protein ligand complex. And we are assuming binding in the micromolar range. So what happens is that we saturate a proton on the protein. When the protein forms a protein ligand complex, this saturation will transfer, this magnetization saturation will transfer to the ligand proton only when they are in the complex. So again, I'll say we exclusively saturate the signal delta B of the protein. This saturation will be transferred to the ligand proton delta A only if they are forming the complex. So the difference in delta A is taken when delta B is saturated and the control case when it is not saturated. We will look at how the spectra appeared. We don't have to worry about the pulse sequence. We'll just look at the spectra. We just talked that protein is taken in small concentration. Picomolar is also fine since we are detecting the ligand. We are detecting delta A, which is on the ligand. So small concentration of protein is fine. If protein is micromolar, ligand needs to be in millimolar. So what we observe in the protein, so suppose delta A is the ligand signal, delta B is the protein signal. This delta B is irradiated. A particular signal in the protein is irradiated until saturation. Saturation means that this peak will disappear because we have studied that in NMR you have energy levels alpha and beta. Saturation means that both alpha and beta are equally populated. So you can see, suppose 2 nuclei in each. This is the saturation case. In this case, there is no net absorption, and so there will be no NMR peak observed. So supplying enough energy to cause this case is what this irradiation is doing, and that will lead to disappearance of the delta B signal. Not only due to this irradiation of the delta B signal, some saturation will be transferred to delta A, and the intensity of delta A will decrease. So that is what is being observed here. And so the peaks we observe are only for the free ligand. We are not observing any peaks for the ligand bound to the protein, because the ligand bound to the protein is getting saturated by this irradiation. So we are saturating delta B, and its peak is disappearing. That saturation is getting transferred to delta A, uh, and only the delta A, which is part of the protein, that peak will disappear, because that is getting saturation transfer from delta B. But the delta A of the free protein, so the free ligand, delta A of the free ligand is far away from this irradiation, so you will still see it. So the intensity of delta A will decrease, but it will still be visible, because a lot of the ligand is still free. So from the free ligand, you will observe a lower intensity of delta A. And this is because initially we had taken a much larger concentration of ligand compared to protein. If we look at the spectra now, so ligand and protein, the signals are distinct. And we have, so suppose so the two signals are distinct, and STD NMR is basically the difference between these two spectra. So spectra A minus spectra B. So what this will do is that you will notice a clear change in intensity of the spectra, and that will only happen if the ligand and protein signals are distinct. Because if they merge, no STD NMR will be observed. You will not see anything in the different spectra. We will look at experimental spectra now. So this is your ligand. Now, this is 1H NMR in the control. That is all the peaks that are observed uh, before we are doing any saturation. So this is the 1H NMR of the ligand. After the saturation, we are only observing DEBF. That is DE, B, and F. Since only these peaks are being observed, these are the atoms that are binding to the protein. So that is how you can remember that in STT NMR, the peaks that are observed, only those regions are binding to the protein. So this will give us an idea of the binding epitope of the ligand to the receptor. So we can accordingly modify the ligand or receptor to get a higher binding, because now we know the binding epitope very clearly. So high throughput drug screening can be done using this method. This is another example. So there are two isomers here. The protein is human serum albumin protein, and you have two molecules, basically tryptophan with a CH3 at the 6th position, or tryptophan with a CH3 at the 7th position. Which of these is binding to the human serum albumin protein? Initial spectra is, so 6 CH3 tryptophan is shown in black, 7 CH3 tryptophan is shown in red. Initially, both are present. In the STT NMR, only the black peaks corresponding to this molecule are observed. So this is the molecule that is binding. So what we saw previously also, the peaks that are observed are the atoms that are binding. So here also peaks of only this is observed. So this is the one that is binding. So using this STD NMR, we can find out which is the binding molecule. How it is used in drug discovery, I will now discuss. So basically, suppose you have a protein target and you have a mixture of ligands that you want to screen to find out which is a good binder to the protein. So then the STT NMR is done, and non-binder, they will show, you can see, if this is the reference 1D NMR of a non-binding, suppose this square, it is not binding. If it gives a control NMR like this and STT NMR, all peaks are gone. That means it is not binding. Or if you have this circle molecule, which is binding, Here the 1D NMR is shown like this, and STD NMR, the peaks will be retained. Intensity might vary, but peaks will be retained. That means it is binding. So using this method, we can screen that which are the ligands that are binding and which are the ligands that are not binding. So STD NMR is used like this in drug discovery. Now we will talk more about drug discovery. So drug discovery is a process that involves many years, many failures, and a lot of uncertainty. Most often, the development of a new medicine starts when basic scientists learn of a biological target, that is, a receptor, enzyme, protein, or gene that they want to target. So, a disease is chosen and there is a drug target. So, suppose a disease is causing due to some protein-protein interaction and we want to inhibit that interaction. So, then that becomes our drug target. Then a bioassay is identified, which is basically a test to determine the biological activity. So since we need to screen a lot of drugs before we find the correct drug, we need a test to determine that how each drug is working, the biological activity of each drug. That is what we are calling the bioassay. Then the pharmacophore is identified. Pharmacophore is basically the structural features directly responsible for the drug activity. Then we can synthesize a lot of possible ligands having those structural features. Then the structure of the drug is optimized to improve interactions with the target protein. And we find a lead compound. Lead compound is a structure that has some activity against the chosen target, but is not good enough to be the drug itself. So once the lead compound is found, it can be optimized further and further till we reach the final drug. This is the basic process of any drug discovery. Better medicines are iterative improvements on current medications. So based on the current medications, better medicines are developed. What do we mean by better? It is better in terms of potency, that is how effective it is against the disease, safety, that is minimum side effects, tolerability, that is how much the body is able to handle the medicine, or convenience, like price or availability. Historically, drugs were primarily discovered by empirical methods, investigating substances or preparations of materials such as plant parts or plant extracts found in the local environment, and then isolation and purification of the active principle in medicinal plants. This was the conventional way of drug discovery. Many such things have been found, like quinine has been found for malaria from plants, or morphine for pain, or hyoscyamine for stomach or intestinal pain, digitoxin for heart failure. These are all naturally occurring classical therapeutic agents found in plants. These biologically active natural products were structurally modified in order to optimize their pharmacology and drug properties. So this was the initial structure of quinine. This had been modified slightly, like this. Instead of closed, it is now open chain, and a chlorine had been added at the 7 position to get chloroquine, and that had been modified further by adding a hydroxyl group here to get hydroxychloroquine. So by improving like this iteratively, the potency and other properties of the drug was improved. And this leads to better and better drugs. It is interesting that the exact configuration of the drug is very important. Both these molecules are exactly the same, except in one carbon center, that is a chiral carbon. In one, the configuration is R, and in another, it is S. When it is R, it is a mild sleeping agent to treat morning sickness in pregnant women. However, when it is S, it causes serious fetal abnormalities. So a single configuration at a single carbon atom can change the properties so extensively. Now, the NMR-based strategies that can identify weak binding compounds, So NMR is initially used to identify the weak binding compounds, then develop them into more potent drug-like inhibitors to use as lead compounds in drug discovery. NMR, we know, can provide structure of a macromolecule at atomic resolution. It can give an idea about molecular and supramolecular dynamics. It can characterize weak interactions between molecules, the protein-protein and protein-ligand interactions, and even for membrane-embedded systems. So these are proteins that are embedded within the molecule, but even idea of these can be gotten from NMR. This we saw a few slides ago, that the NMR properties of ligand and protein are completely different, but protein and protein ligand complex have similar properties since they are both large molecules. So for investigating the physical chemical principles, one thing is chemical shift mapping. So protein ligand, protein nucleic acid, and protein protein interactions are on primary interest in structural biology and drug discovery. NMR can characterize both high and low affinity macromolecular complexes. And so that is an advantage, that from nanomolar to millimolar, NMR can characterize all the affinities. In some cases, you might need to prepare samples with stable 15N or 13C isotopes. Now, what is chemical shift mapping is that binding of a ligand or macromolecule like nucleic acid or protein will alter the chemical environment around the binding site. So if this is the free protein, when it is binding to another protein, this binding site shown in red, the configuration of that will change, the chemical environment of that will change. This will perturb the chemical shift of the nuclei at this site. So here, if the original position of this leucine 65 was at green, then as it is binding, it is shifting more and more away from that. So leucine 65 must be in the binding site because its chemical shift is changing so much. But leucine 44, it remains in the same place, so it's not part of the binding site. So this change in chemical shift can be observed using 1H15N or 1H13C HSQC spectra. Here what is shown is 1H15N. Now, these are typically for small proteins like up to 100 to 200 amino acids. More than that, HSQC spectra might become more complicated. So in if it's a small enough protein, you will be able to see the shift in 1D spectra also. So you can see here a shift in one of the tryptophan protons is shown. The red spectra is in absence of the ligand, the blue spectra is in presence of the ligand. So wherever shift is there, that indicates that is involved in binding. And if there is somewhere where there is, so you can see this peak, there is no shift at all. So it is not involved in binding. Similarly, peaks can shift in 2D peaks will give an idea about the binding site. So here, this peak before binding has shifted here after binding. Similarly for this peak. So these are involved in binding. However, these peaks, either the shift is very small or it is completely absent. So they are further away from the binding site or they are not at all near the binding site at all. So this chemical shift changes will give an idea about the binding. Here, now what if it's a large protein like dipicolinate reductase, DHPR, which is a 120 kilodalton homotetrameric protein. Now here, we know that out of the 20 naturally occurring amino acids, 6 of them have a methyl group, and we can use these as probes. So methyl groups can exclusively be labeled by feeding selectively labeled compounds to bacteria and using their metabolic pathway. So when bacteria which are fed on selectively labeled compounds, they will produce proteins where only the methyl group in the 6 amino acids are labeled. And we know methyl groups are in this particular protein, methyl is distributed all over the protein. So if you get an idea of the methyl groups, you will get an idea of the structure of the protein. It will also considerably cut down on the NMR crowdedness because we are not looking at the other amino acids. We are only looking at the methyl groups. So here, different methyl groups are shown in different color. And this is also a HSQC spectra, 1H13C, because we are looking at a methyl group. So from this, we can get an idea of the structure of the protein. Since the methyl groups are more or less distributed all over the protein, getting an idea of the position of the methyl groups will give an idea of the structure of the protein. Now we will look at NMR-based drug discovery strategies. The first one is SAR by NMR, where SAR stands for Structure Activity Relationship. SAR by NMR is based on the use of protein chemical shift changes to screen for low affinity ligands and the collection of structural information to direct a linked fragment approach to enhancing binding affinities. So this is a ligand-based method where the ligand binding is detected by the chemical shift mapping. So suppose there is a protein with two binding sites. We will first screen different possible ligands for the first binding site. Suppose we have a ligand with relatively low binding, 17 millimolar, and then different ligands will be screened for the second binding site, which also we get a low binding ligand of 20 micromolar. Both these, whether it is binding or not, is determined by the chemical shift changes. Then the next is basic. So here the example shown is for human stromylysine, this protein. And so we saw for the first binding site, one compound was found to bind around 17 millimolar. And for the second binding site, a compound was found to bind around 20 micromolar. So we screened weak ligands and then structurally directing the chemical linkage of two low affinity ligands. So these two low affinity ligands are chemically linked by finding a linker of appropriate length and structure to create a combined ligand which has high affinity. So this combined ligand is showing affinity in the nanomolar range. So this method by which protein chemical shift changes are used to screen for low affinity ligands. So individually, these are low affinity ligands. And we get an idea of the structure that is fitting the active site. And then a linked fragment approach is taken. So these fragments are linked to enhance the binding affinities, which we can see from millimolar or micromolar individually, it has gone to 15 nanomolar. A strategy used in this is called the shape screening. Here we assess the binding of a fairly small but diverse library of low molecular weight scaffolds to a drug target using NMR techniques that are based on observing the ligand. So like SAR by NMR, this also it is looking at the ligand. There is a small but diverse library of these low molecular weight possible ligands or scaffolds. These are mainly derived from shapes or frameworks that are commonly used in known drugs. Initially, weak binding hits in the millimolar range have been used in several follow-up strategies. Initially, you can see the binders are weak binders like millimolar. Then slowly, as it is these hits have been used as starting platform for virtual screening of compound analogs to guide medicinal chemistry. On the basis of this initial weak finding, a library of analogs with further derivatization of the initial core structure was screened to obtain further higher affinity ligand. So the initial core structure is there, one 5-membered ring, one 6-membered ring, that is derivatized to get a further library which has comparatively higher binding micromolar. Then further substitution of this higher binding affinity ligands resulted in compounds that are very potent inhibitors. 1D NMR screening was used to identify higher affinity ligands of the weak binding hits. So again, further derivatization was done. In the initial derivatization, it was two 6-membered rings attached to a 5-membered ring. Then a third 6-membered ring was introduced, and it was seen the Kd goes to nanomolar. So these are now very potent inhibitors. This entire strategy where a small but diverse library. So the libraries created are small, but it is pretty diverse to account for all shapes, are screened against a drug target using NMR techniques. And this process is repeated. This is called shape screening. Another technique is NMR solve, where solve stands for structurally oriented library valency engineering. This is a drug design strategy that provides information for the construction of focused libraries against multiple related target. So in shape screening, ultimately these libraries that we were forming were random. We were randomly derivatizing to find better and better hits. In NMR solve, the library is formed based on the information of the binding site on the protein. And it is not against a single protein, it is against a representative member of a protein family. So protein family means that they have common bindings. It exploits the fact that large families of proteins have adjacent binding sites, one of which is conserved throughout the family. So the first binding, this is a family of say N proteins, where the first site is conserved in all the proteins and the second one is varied. So taking any one representative member of the family, we selectively label the amino acids around the binding sites. These are the probes around the binding sites. And this is a probe between the two binding sites to find out what kind of linker should be there. So these probes tell us in how will we find out which is the binding site is that subtle chemical shift changes in the reference ligand on the HSQC spectra of the protein will tell us about where is the binding site of the protein. And there certain probes are introduced, and also between two binding sites, a probe is introduced to find out the orientation between the two binding sites. So now the library that is used is a focused library because we have an idea about the kind of structure of the receptor site. So instead of a random library that was used in shape screening, it is now a focused library, and it is against multiple related targets, that is proteins within the same family, because they all have the first same binding site. Once a low binding affinity ligand is found for the first site, the orientation of a common ligand mimic in the binding site is determined to provide a basis for the design of this linker, which is directed into the specificity site. So this is called the binding site because it is same in all the members in the protein family. This is called the specificity site because it is distinguishing between the members. So this probe will tell us that what should be the orientation of this linker joining the receptors of the two binding sites. The NMR, so eventually that is also optimized. So we saw in SAR by NMR how two ligands can be linked in order to get a much more higher affinity ligand. NMR solve is a method of SAR by NMR where instead of blindly designing the ligand, the orientation is found using a probe so that the ligand can be more accurately designed, and it is done for the entire family of proteins. So the NMR data of protein 1 is used to guide the synthesis of a combinatorial bi-ligand library, which is screened in activity assays against protein 1 and related proteins. So we are getting NMR data of protein 1 using these probes, and we can use this to guide the synthesis of a combinatorial bi-ligand, meaning two ligands linked up with each other, and they can be screened not only against this protein but against the entire family of proteins. So this kind of method is called fragment-based drug discovery, because we are discovering two separate fragments that are binding with two different sites, and then we are linking them. And how we are finding out? It can be 1H, 15N HSQC from the chemical shifts. So then the protein will need to be 15N labeled. And then the linker is optimized to get the biligand libraries. Another way in which the ligand is, the linker is designed is through ILOE. So if you have two ligands which are binding to two nearby binding sites, the protons of the two ligands will show NOEs, which are defined as ligand-ligand NOE or interligand NOE, that is ILOE. So using this, it will give you an idea about how the linker between these two ligands can be constructed. So that is the advantage of these ILOEs. Just 1 second. Just 1 second. So what are the basic properties that a drug should have? It is summarized by ADME, that is absorption. The drug should be properly absorbed in the body. Distribution, it needs to be distributed throughout the body so that it reaches at the place it needs to go. Metabolism, the drug must be broken up in the body, otherwise it will just accumulate. And excretion, ultimately the side products must be excreted so that it doesn't accumulate in the body. Another point, of course, is the toxicology studies, that it cannot be toxic to our body. Physicians prescribe drugs on the basis of the characteristics of the medications and on the probability that reliable and reproducible clinical results will happen. But the problem is there is a difference in drug response among patients. So this leads to challenges in optimizing a dosage regimen for individual patient. Another problem is that majority of drugs are effective only in 25 to 60% of patients. So why is that? NMR has been used for the study of this drug metabolism in vitro, that is in biofluids, tissue extract, biopsies, intact cells, perfused organs, and in vivo, that is within the animal or human subjects while they are still alive, that is MRI. NMR spectroscopy is unique in its ability that it permits both these kind of studies, in vitro or in vivo. If we look, we are looking at an example protein, cytochrome P450 enzymes or CYP, which are important in biosynthesis and degradation of endogenous compounds, such as steroids, lipids, and vitamins. The expression of this protein can be different in different persons. So this protein is causing the degradation of these compounds. So since this is expressed differently in different persons, so if you're taking a drug, that will also act differently in different persons. So metabolism of the drug is different, and this results in a difference in availability and efficacy of the drug from person to person. About 40% of commonly used drugs is bioavailable because of limited absorption. That is the first pass metabolism. So if you're taking, say, 100% of a drug, only the 40% will be of use. The rest will just be excreted by the body. And this will also vary from person to person because metabolism of each drug is different from person to person. As you know, some people have very high metabolisms. No matter how much they eat, they don't gain weight. So metabolism is different in persons based on their genetic makeup or other things. And that will result in different availability and efficacy of drugs from person to person. So that is one of the reasons why the dosage in people can vary. And also they are effective only in a certain percentage of patients. Now, the liver is a major site of this cytochrome P40 mediated metabolism. And another potential site is the enterocytes in the epithelium of the small intestine. This basically, it is this protein that is metabolizing the drugs that is causing the problem, because this protein is responsible for drug metabolism, and it is expressed to different extents in different people. So metabolism in people varies due to that. And if suppose someone is metabolizing the drug very fast, so it is active in their body only for a short time, or someone is metabolizing it very slowly, so it is active in their body for a longer time. That is what causes the difference in dosage or efficacy of a drug from person to person. Now, with metabolism, the bioavailability of the drug will decrease. This determines the dosage of the drug. We will look at a drug, felodipine. Suppose we are taking initially when it is entering the stomach, it has 100% dose. In different regions, dosage will decrease. Like you can see here within the intestine, dosage has decreased 45%, 15%. So like this, depending on how much it is metabolized, the dosage decreases. And according to that, we need to adjust the initial dosage. Felodipine is generally used to treat high blood pressure. It relaxes the blood vessels so that the heart does not have to pump so hard and the blood pressure reduces. Now, since the availability is decreasing, an important research area is how the bioavailability can be increased so that the dosage does not need to be increased too much. Studies have shown that this is the felodipine concentration if the drug is taken with water and if the drug is taken with grapefruit juice. Concentration of drug in grapefruit juice increases up to 30, say around 25%, even after. So if you're taking water, the maximum concentration of the drug is only around 10%. But in grapefruit juice, it goes up to 27, 28%. And even after 8 hours, the water bioavailability has almost gone to zero. But in grapefruit juice, it is still bioavailable. So just depending on what you are taking the drug with, the bioavailability will change. Hence, you can see that increased concentration of drug in grapefruit juice results in more efficient lowering of blood pressure. So here blood pressure is shown, and if you take here the number of hours after which the drug has been taken. Since in grapefruit juice the bioavailability of drug is higher and it also stays longer, the reduction in blood pressure is more efficient when it is taken with grapefruit juice. So the bioavailability of the drug is a very important point that determines how effective the drug is in treating the disease. Now, one thing to target is the cytochrome P40 or CYP protein that is metabolizing the drug. If you can inhibit the protein metabolism activity, then the bioavailability can be increased. So in healthy subjects, suppose the bioavailability of this felodipine is shown, but there are certain patients who have a disease which increases the CYP activity. That means the drug metabolizes very fast and bioavailability is very low in these proteins. It is less than 1 and it goes to 0 after 6 hours. Whereas in the healthy subjects, the drug is bioavailable even up to 12 hours. So if this activity is increased, the bioavailability decreases. Using the opposite logic, if we can inhibit the activity, the bioavailability can be increased. Now, how NMR spectroscopy can be used to study this drug metabolism? NMR is a non-invasive technique for studying drug metabolism. As we know that NMR the magnetic field is very weak, so it does not destroy the sample which you are studying. So it is a non-invasive technique. It has been used to the study of drug metabolism in vitro, that is in biofluids, tissue extracts, and so on. And the key fluids that are used to study the drug metabolism are plasma, serum, urine, or saliva. It has also been used in vivo in live animal and human subjects. So that is a specialty of NMR. It is unique in its ability to permit both in vivo and in vitro studies. Uh, NMR can also be used in specialized locations like cerebrospinal fluid, pancreatic fluid, amniotic follicular fluid, and so on. These are also in vitro. Now, the isotopes used in the study of metabolism of drugs by NMR, they can be, we will, any NMR active nuclei, but we will individually look at each nuclei and see their advantages and disadvantages. 1H we know has very high natural abundance, 99.98%. So a minimum concentration of the sample is enough for 1H NMR. So proton, the advantages are it is very abundant. We just discussed 99.98% natural abundance. So a minimum concentration of the sample is enough for the proton NMR. And also a good thing is that it is present in all drugs. And so it has the highest NMR sensitivity. Even a small amount will give a peak of large intensity. But the problem is that proton has a small chemical shift range, only up from 0 to around 15 ppm. And there is extensive multiplicity due to homonuclear J coupling. That is 1H1H J coupling will cause the spectra to split. There is a lot of multiplicity, triplet, quartet, and so on, which causes the spectra to get crowded. This makes the observation and quantification of drug metabolites difficult. Another problem is that 1H spectra of biofluids or tissue contain many intense resonances from water. We know a lot of our body is water, so all these biofluids will have peaks from water also. They will, for that, water suppression methods need to be used like pre-saturation, gradient-based freeze drying, etc. But there are also additional 1H peaks from proteins and lipids. Basically, 1H is a very common nuclei. It is occurring in a lot of the naturally occurring molecules in our body as well. And that needs to be reduced or eliminated so we can focus on the drug metabolism that we want to study. So that is a disadvantage of 1H NMR. And what about in vivo? 1H is a poor nucleus for the in vivo monitoring of drugs and metabolites. The first thing is that in vitro, you can spin the sample to get sharp peaks. But in vivo, it is not possible to spin the sample. So the lines are broad. And there is a lot of coupling in 1H, which leads to further crowding in the spectra. Also, there is restricted molecular mobility. Unlike in vitro samples, you are not able to spin the person or animal that is the in vivo studies. So there is restricted molecular mobility. Hence, magnetic field inhomogeneity happens over the large sample volume. And we also need to use, and we know magnetic field inhomogeneity is one of the reasons for line broadening. So the lines will be broad in vivo. And we need to use low magnetic field spectrometers so that the human body does not get affected. So in vivo line widths are substantially broader than those obtained in vitro. This is a problem in vivo, not only with 1H, but a general problem in vivo NMR. Just give me 2 minutes. Right. So now let us look at the other nuclei. One is fluorine 19. Fluorine 19 has a lot of advantages. Like 1H, it has a spin of half and it has relatively narrow lines. It has an abundance of 100%. So again, similar to 1H, a small sample is enough. So it has high sensitivity. Uh, it has a large chemical shift range. So a good separation of peaks. That is an advantage over 1H, which has a relatively small chemical shift range. And it has short longitudinal relaxation time, that is T1. So the experimental time reduces. And that allows us to improve the signal to noise ratio by scanning the sample multiple times. Another advantage is that a number of fluorinated drugs are currently in use. So these are some examples shown of fluorinated drugs used for different disorders. So 19F offers a powerful method to monitor the pharmacokinetics and metabolism of these drugs because 19F does not naturally occur in the body. So the 19F signal of the drugs will not be confused with any signals of molecules naturally occurring in the body, and you will be able to accurately follow the metabolism of the drug. Unlike 1H, which signal can get confused with the large water content in the body and also the 1H that uh, uh, is there in naturally occurring proteins and lipids in the body. In 19F, there will be no such overlap. Now about 13C, an advantage is the large chemical shift. We know 13C can go from 0 to 150. So the peaks are well separated, which helps in structural identification. The problem is that the magnetically active isotope 13C is not abundant nor particularly sensitive. If we look at the abundance, 13C is only 1.1%. So out of 100 carbon atoms, only one atom will be 13C, rest will be 12C, which is not NMR active. And we know that the drugs cannot be isotopically labeled for practical purposes. So it is not like we can add more 13C. Its abundance is low and hence sensitivity is low. Partially, the sensitivity can be improved with polarization transfer experiments. We have seen this, but the use is still very limited because of its very low abundance. 31P, on the other hand, is extensively used because, again, it has an abundance of 100%, so it is very sensitive. But the problem with 31P compared to 19F is that there are many endogenous phosphates and derivatives. We know all DNA has phosphodiester bonds and so on. So these may interfere with the signals from the phosphorylated drugs and their metabolites. So there is no 19F containing molecules in the bodies, but there are a lot of 31P containing um, atoms in the body, and they can interfere with the drug signals. Also, phosphorus containing drugs are fairly rare. Unlike 19F, where there are a number of drugs that contain fluorine, phosphorus containing drugs are fairly rare. So that is a disadvantage of 31P compared to 19F. Now, for traditional drug metabolite analysis, traditional methods include HPLC, gas chromatography, mass spectroscopy, so on. They require minimum volume of sample compared to NMR. However, these methods can be perturbed by column matrix, retention time, solvent used. So there are a lot of external factors that are perturbing these methods. Also, other problems are they require separation followed by identification. So in HPLC, first, uh, you have to separately collect each peak, and then using mass spectroscopy or other method, the peaks can be identified. So two experiments will have to be coupled. Then the separation conditions have to be optimized for each compound or drug. Often multiple separations may be required, and it is a very slow and manually intensive process. You have to constantly supervise the experiment, and you need enough skill to do it. So these are all advantages with these traditional methods of drug metabolite analysis. The advantages of NMR based method: it is a high throughput method, it gives high resolution, there is least effort on sample preparation. You just need suppose we are doing in vitro NMR using plasma, so 300 microliter of the plasma and 300 microliter of the buffer, and either it has we have to ensure that there are no bubbles that interfere with the spectra, and this is the buffer composition. So that is it. It just needs to be put in a tube and the spectrometer will take care of the rest. So the effort on sample preparation is very minimum. Then it is a quantitative method. And we know NMR is non-destructive because the magnetic field used is very weak. All the metabolites are detected simultaneously in a single analysis. And NMR gives both structural and quantitative information. And little or no spectral evidence from endogenous molecules. In 19F, 31P, there is some spectral interference, but it is still less than 1H, 7 lithium and 13C. So that is another advantage. For NMR spectroscopy, basic things that are needed are a 500 or 600 megahertz spectrometer, a 1D 1H gradient enhanced noisy pre-saturation with mixing time of 10 milliseconds for suppose we are analyzing urine here, which is an in vitro, to look at the metabolites in urine. And these are other pulse sequences. We don't need to go too much into that. These are the settings of the NMR spectrometer basically. We don't need to go into details of that. Here an example is shown of NMR used in drug metabolism studies. For the drug, anti-inflammatory drug, flurbiprofen. So the NMR is shown 600 megahertz 1H NMR spectra in a sample of human urine collected after the oral ingestion of 200 milligram of this drug, flurbiprofen. So again, from the peaks, it can be understood that whether suppose this has broken into different compounds. By identifying these compounds, we can understand how it has metabolized in the body. So from this is another example where it is taken after 1 hour of oral administration of this drug, ethoxyphenacetin, to a healthy patient, what happens, and to a patient with acute hepatitis, what happens. So in a diseased patient, you will be able to see that the metabolite concentrations are different. For example, the 67.5 ppm peak corresponding to phenacetin is quite low in a healthy patient, but it is much higher in a patient with acute hepatitis. So the peak intensities are changing and additional peaks are also appearing. You can see here there is here you are not seeing additional peaks, but the peak intensities of all these are changing. So between the control, that is a healthy patient, and a patient with acute hepatitis, the drug metabolism is quite different because the peak intensities corresponding to the different metabolites are different. So that will give you an idea that the patient has this hepatitis. So in vivo studies of drug metabolism, the advantage of this is that if measuring the drug levels in plasma does not always affect the drug concentration at the receptor site, because plasma is not being withdrawn from the receptor site, it is just being withdrawn from the blood. That can be anywhere. And so in vivo takes care of that because you are directly measuring the drug and the metabolites in the target organ. It is a method that allows one to measure the concentration of drug and their metabolites in situ, that is in the target organ itself, which is very useful. In vivo, it can be used repetitively on the same patient. And it is non-invasive because we use a low strength magnetic field. The drawbacks, however, the sensitivity is much less than in vitro, 1 to 10 times less. Because in vitro, you can increase the strength of the spectrometer to get a more and more well-resolved spectra. But you cannot put the human being inside too much of a high field spectrometer. Otherwise, there can be problems. Another thing, molecules have only molecules with unrestricted mobility are visible. So since we cannot move or spin the human body, there is a tendency for line broadening. So you will only see molecules where uh, there is a lot of mobility. Prolonged data acquisition may be required to make up for the sensitivity. The observed signals may have various anatomical origins. For example, if we are doing 1H, we can get 1H signals from the water in the body or from the protein or lipids that are naturally present. So these are anatomical origins. So the observed signal may not be from the drug that we want to study, but it can be from different other naturally occurring anatomical regions. So the resolution is poor because of this low sensitivity and line broadening. So chemically similar compounds will be difficult to distinguish. So these are the drawbacks of in vivo over in vitro. Now the application of NMR in drug metabolism studies. 19F NMR studies are related to anesthetic, psychoactive, and anti-neoplastic drugs. So we have seen 19F is found in a lot of drugs. So these are some examples of those drugs. The distribution of this anesthetic in the brain and their pharmacokinetics of elimination are still a subject of controversy. It has not been fully elucidated so far. The feasibility of in vivo 19F NMR studies of halothane in humans has been demonstrated. And fluoxetine is also a drug containing fluorine, this fluoxetine, that has also been studied both in vivo and in vitro. In vitro, you can see the peaks of fluoxetine and one of its metabolites, norfluoxetine, are very sharp. In vivo, the peaks are much broader and they are also shifted. And you can see signal noise ratio is not very good in vivo compared to in vitro. 19F studies on fluorinated drugs with 5-fluorouracil. So in this 19F studies, high doses are administered to patients. The fluorine atom remains intact during biotransformation. So no matter how the drugs are metabolizing, the fluorine atom remains intact. And they are displayed over a large spectral width and there is no peak overlap. So these are advantages of 19F. And you can see that different metabolites will clearly appear up in the 19F spectra. And because of the large spectral width, peak overlap is minimum. And also the fluorine atom remains intact throughout the biotransformation or metabolism. So you will be able to study its journey throughout the body. This fluorouracil is injected, it goes into the body and gets metabolized. There are different products formed in the metabolic pathway that can be detected by NMR. These have been labeled here and these can all be used. This is from a perfusate of body weight, so it's in vitro, and these can all be used to understand the metabolic pathway of the drug. So in vivo 19F NMR has been used to monitor fluorouracil metabolism in the liver. In colorectal cancer patients, they were treated with a low dose intravenous infusion of fluorouracil until the point of refractory disease. So from the 19F spectra of liver metastasis of the patient who was treated with this fluorouracil drug at a dose of 300 milligram per day, the NMR was studied. This fluorouracil Fu is getting converted into a catabolite. This is A-fluoro-B-alanine. We don't need to look into that. So basically, you can initially, suppose this is after a few hours, you have the concentration of catabolite, but you also have fluorouracil. So the drug is still bioavailable. So if this is after, say, 2 hours, after 10 hours, the concentration of the drug is completely gone, and you have only the metabolite peak. So that will give an idea of how long the drug is bioavailable. Now, if something else is added, like that is interfering with the fluorouracil activity, then in that case, even after 10 hours, there is enough of a concentration of fluorouracil. So that is reducing the metabolism of fluorouracil. So suppose you, in a patient, you need a very acute dosage. You need the fluorouracil to immediately get metabolized. In that case, you will not add the interferon. But if in another patient you need a lower dose of fluorouracil over a prolonged period of time, you need it to work slowly, then you will add this interferon. In this case, the metabolism of fluorouracil will be slowed down, and it will persist in the system even after a long time. Now we know that we know about magnetic. So MRS is a method that is used in vivo to detect various metabolites. Uh, Imagine in a mouse brain, without the presence of a magnetic field, all the spins are randomly oriented. When you place the mouse in a magnetic field, uh, with a specific radio frequency, all the spins will get oriented, either aligned with the field, which is lower energy, or opposite to the field, which is higher energy. Uh, this radio frequency that the field is providing, it will bring the magnetization, it will turn it by 90 degrees. We have studied this, and this will be detected as the NMR signal. So and slowly, as the magnetization will go back to its original position, the NMR signal will decline, um, and the system will go back to its original position. So this is NMR spectroscopy, but in vivo, and this has been given the special name of magnetic resonance spectroscopy, MRS. This is basically like the 1D NMR spectra itself, and various metabolites can be detected using this method. Uh, so MRS is what we looked at right now. MRS spectra is basically like a 1D NMR spectra, and you can detect various metabolites from it. MRI, which is the more commonly used method, it is comparable to the 2D NMR spectra or 3D NMR spectra, where you will get an idea about cross-sectional image of the organ you are looking at. So you can look at the organ in two-dimensional cross sections. So it will give more information. This basically is comparable to 2D NMR, and the machine will be able to convert it into the cross-sectional images, which will clearly show whether any tumor or other defect is present. Whereas this will give different kind of information. It is a 1D spectra, the MRS, which will show the various metabolites, and we can compare that with a healthy patient, and if the levels are different, that will indicate that some disease is there. So this is the difference between magnetic resonance spectroscopy, MRS, and magnetic resonance imaging, that is MRI. So here an example is shown of 31P MRI and MRS of human liver. So MRS is basically giving this one-dimensional spectra, and it is showing different metabolites like ATP, NADPH, and so on. If this was the baseline MRS, this is after a seizure. So after a seizure, the energy currencies like ATP and NTP, they are higher compared to baseline. So this means that after a seizure, the brain is becoming hyperactive. So MRS, that is giving an idea of the metabolites in a normal case, that is the baseline, and after a seizure has taken place, and the difference in intensities of certain signals will tell us that indeed a seizure has taken place. This is MRS from the cortical region of a patient who has a brain with Alzheimer's. So in the wild type, there are certain peaks whose intensities are changing in this Alzheimer's patient. NAA peak intensity is decreasing, GLU peak intensity is decreasing, and there are certain new peaks of new chemical compounds that are forming in the Alzheimer's patient. This gives very useful insights into how the disease is changing the metabolic state of the brain. So comparing the MRS spectra of the healthy patient and the diseased patient will give us an idea about that. Just 1 second. Before we go into the question answer part, I'll just just 1 second, okay, before we go into the, I'll go into a few more concepts before we go into the question answer part. I'll talk a bit about protein dynamism because I mentioned that, but I did not go into the details of that. So a protein can have many different kinds of dynamics. It can have local flexibility, which is bond vibration, methyl group rotation, motion of a loop, or the side chain can exist in different rotamers, or it can have collective motions. So suppose there are two domains, one domain is moving with respect to the other, that will be the collective motion. And these are all of different time scales. The local motions occur at lower time scales like femtosecond, picosecond, whereas the larger domain motions occurred from millisecond to second time scale. And there are different methods to study the different motions in the protein. NMR spectroscopy can study anywhere from second to picosecond range motions. And you will be able to identify most of the motions using NMR spectroscopy, except some bond vibrations, which can be studied using IR. And sometimes NMR spectroscopy is coupled with hydrogen deuterium exchange experiments to capture the slower time scale motions. Now, the problem is the other methods like fluorescence, UV vis, they do not have as much resolution as NMR. So NMR can not only give an idea about the time scale of these motions, but it can do so at an atomic resolution. And that is the advantage of the NMR. Now, these are some other experimental methods that are used. The problem is that in X-ray crystallography, you need a homogeneous crystal. So many compounds don't crystallize at all. For those, X-ray crystallography can't be used. And also, in X-ray crystallography, you are locking the molecule in a particular conformation. In NMR, it is in solution, so you're allowing the molecule to adopt different conformations, and so it is a more accurate picture of the actual case. Whereas in X-ray crystallography, you are trapping the molecule in a particular conformation, and so it is not that accurate. Cryo-electron microscopy is an emerging technology, but it is suitable only for bigger molecules. Then there can be small angle X-ray scattering, but the problem is this will not give atomic resolution information. And free electron laser methods are also there, but it is a high powered method which will destroy the sample. So sometimes two or more methods are combined to get accurate information. So NMR relaxation will give an idea about protein dynamics. the fast motion in picosecond to nanosecond time scale, they can be obtained from, say, there are different experiments for these fast motions like R1, R2, the relaxation methods, NOE experiments for this fast motions like back change in the backbone dynamics, which occurs in picosecond to nanosecond time scale, or rotation of a side chain that occurs again within the picosecond time scale. These can be detected using different NMR experiments like relaxation experiments or NOE experiments. The uh, slower, if you AHT exchange can tell you about the formation of secondary structures like alpha helix or beta sheet, which form in a nanosecond to millisecond time scale. And the slowest, that is protein folding, which occurs from millisecond to hours time scale, that can also be found from HD exchange experiments. And there are these are two kinds of motion. The fast motion is from picosecond to nanosecond, and slow motions are from microsecond to millisecond. And extremely slow motions, millisecond to second, there are special NMR experiments called magnetization exchange spectroscopy or ZZ exchange. We don't need to go into the details of this. You can just remember there are different time scales and different NMR experiments to understand the motions in each time scale. Now, in the fast timescale motions, they occur in the local minima of the energy landscape, whereas in slow timescale motion, the energy goes from one state, the protein goes from one state to another state, that is a global motion. So A is a global minima, B is a local minima. Suppose the protein goes from B to A, that is, there is a huge conformational change in the protein. This occurs on a slower time scale, that is microsecond to millisecond. This this kind of conformational change occurs in enzyme catalysis or signal transduction or protein-protein interaction. These are very large-scale motions or global motions which occur on a slow time scale. And so within this particular state, you have a range of small energy fluctuations. This is where the local motions are occurring. So again, within this state, there are small energy fluctuations. This is where the local motions are occurring. Since the energy barrier in this local motion is very low, there is not a lot of, here the energy barrier is so high, but in the local motion, energy barrier is so low. So just with thermal fluctuation, many of these states can be sampled. Whereas for global motion, you might need to provide energy to the system. So these are basically studied by relaxation studies. Again, we don't need to go into the details of this. I'll come to the experiment itself. I'll come to the experimental observation itself. We don't need to go into the experimental details, so I'll directly come to the observation. So suppose you have a protein here. The terminus of the protein, the N and C terminal, are very flexible. And there are, in the core of the protein, it is very well folded into alpha helix, beta sheet, and so on. So R1 is 1 by T1, where T1 is the relaxation time and R1 is the relaxation rate. So one thing you can just remember is R1 is high for flexible domains. That is N and C terminal, these residues will have high values of R1. You can see on the x-axis, you have residue number, and on the y-axis, you have R1. these residues near the N terminal and this residues near the C terminal, they have high R1. So these are flexible residues in the N and C terminal. Whereas in the core of the protein, they are well arranged into secondary structures. They will have low R1. You can see all these residues, they have low R1. Sometimes within the core, some residues have high R1. So within this is representative of loop region. You can see two beta sheets are connected by a loop, or alpha helix and beta sheet is connected by a loop. These loop regions are quite flexible and hence they have high R1. So R1 is telling you about the dynamism of the system. High R1 indicates that region is very dynamic. It can either be a flexible domain like the N or C terminal, or it can be a loop which does not have a specific conformation. Whereas low R1 indicates that the conformation is very restricted. These generally refers to well-folded secondary structures. Now, when you add urea to the system, urea basically denatures a protein. So from folded, it goes to unfolded. You can see when urea is added to the protein, most of it becomes unfolded. And so almost all the residues, they have a mean value of R1. So here the R1 was changing quite a lot. Now the R1 is more or less same for all the residues because the protein has become denatured. It is not, it does not have a specific conformation anymore. So that is what this urea is doing, is that most of the R1 values are near a mean line because it is losing all secondary structures. So that is also very unique, that when the protein unfolds, the R1 shows that. Now there is another relaxation time, which is R2. So we looked at R1. So one is based on the spin relaxation and one is the spin lattice relaxation. R1, we saw that the flexible regions had high R1 and the secondary regions that were confined in a secondary structure, they have low R1. R2 is the exact opposite. So in R2 is the reverse of R1. Flexible portions, that is N and C terminal, they have low R2 values. So N terminal, low R2, C terminal, low R2. And in between, there are the loop regions, they also have low R2. And the rigid regions that are confined in a secondary structure, they have high R2 values. So R2 is exactly the opposite of R1. But in 8 molar urea, it behaves similar to R1. We saw that in 8 molar urea, all the residues have similar R1. Similarly, in 8 molar urea, all the residues have similar R2. So using this, it will give you an idea about whether the protein is like dynamic or it is like it will give you an idea about the flexibility of the protein. So that is about the different molecular motions in the protein. So that is a concept I wanted to discuss. Now I will go into the sample questions of the class we were discussing. The first question is asking which one of the following statements is true for in vivo NMR. So here the answer is D. So if we look at in vivo NMR, the advantages were that you are measuring the drug and metabolites in situ in the target organ itself, and you can repetitively use it on the same organ, and that it was non-invasive because we are using weak magnetic fields. The drawback, however, is that the sensitivity is very low. So that means sensitivity is high is incorrect. So if sensitivity is low, resolution will also be low. So this is incorrect. Another is that, as we talked about, the in vivo, you cannot spin the sample in order to remove field inhomogeneities. So because of that, instead of a sharp peak, which we observe in vitro, in vivo, the spectra will be broadened. So that will again affect resolution. So another problem is that to improve the sensitivity, the data may need to be acquired for a longer time, and the observed signals may have various anatomical origins. So in vivo, we have, suppose we are for 1H, there can be signals from water because 70% of the body is water. There can be signals from 1H occurring in naturally occurring proteins and lipids in the body. So the signal is originating from various sources, not only from the labeled probes. So this is also wrong. And resolution is poor, that we have already discussed. So the data acquisition needs to be extensive. That is this point. Data acquisition is prolonged because you are having low resolution, low sensitivity, line broadening. In order to make up a little bit for all these, the data acquisition needs to be taken for a longer time. So option D is correct. The next question is asking, why is 19F a good probe for in vivo NMR? So here, if we look at the favorable characteristics of 19F, it remains untransformed during metabolism. So it remains intact during biotransformation. Basically, since it remains untransformed, you will be able to study its entire journey from the time of ingestion to excretion, since it remains untransformed. So this is a positive point. Peak signals have a large spectral width. We have already seen that large chemical shift range. So signal overlap is minimized. That is also a positive. It has high natural abundance, 100% natural abundance. So even a small sample is enough for a high intensity signal. So all these are positive points. Other than that, other positive points is that there are a large number of fluorinated drugs currently in use, and 19F does not naturally occur in body, so it will not overlap with signals from anatomical origins like there was in 1H or even 31P, because there is phosphorus in DNA or RNA. So unlike that, 19F does not have overlap with any signals from the body. So these are all positive characteristics of 19F. A, B, and C are all reasons why it is a good probe. And so the correct answer is D, that is all of the above. The next question is asking, looking into the limitations of spectra obtained from in vivo conditions, which probe is generally used? So here, if we look at the different options, 1H, the problem is that it has a small chemical shift range only up to around 15 ppm. So there can be a lot of signal overlap. Another, due to the 1H 1H J coupling, each signal is split up into triplets or quartets or even more multiplicity, and that will lead to even more overlap, making the resolution very difficult. Also, in 1H, there can be confusion with the water signals from biofluids or signals from protein and lipid that naturally occurring in body that also contain 1H. So these are a lot of limitations of 1H. 14C is NMR inactive. We saw that if the mass number and atomic number are both even, the nuclei is NMR inactive. And we know 14C, the atomic number is 6. So both mass and atomic number are even, so it is NMR inactive. 235 uranium is radioactive. You cannot use a radioactive isotope, it will harm the body. 31P, on the other hand, the advantage over 1H is that it has 100% abundance, and so signal is very sensitive. That is same as 1H, but more importantly, the compared to 1H, it has a larger chemical shift range and also multiplicity is less. So that makes 31P a very useful nucleus compared to the other given options. If 19F was an option, then we would have chosen 19F since it is better than 31P, because 31P, the problem is that there are not a lot of drugs that contain 31P, but 19F is there in a lot of drugs. And also 31P can have some interference from the DNA or RNA phosphorus signals, but there will be no interference in the 19F signal. So if 19F was an option, we would have chosen that. But in the given options, 31P is the best. So that is what we are choosing. That is option D. The next question is asking samples for assessment of NMR specific drug metabolism can be which of the following? So here the answer is urine. So if you remember the key diagnostic fluids, they include plasma, serum, urine, saliva. Sweat is not included because sweat is mainly just water and some salts. You will not see any metabolites in sweat. So that is out, which means automatically all of the above is out. And hair is just a keratin protein. It will not show the drug metabolism we want to see. It is not a living part of the body, that is where the metabolites will go, and so hair is out. So among these options, urine is a correct option. So the correct answer is B. The next question is asking which of the following options indicate NMR isotopes having a spin value of half. So here, if you look at the list of common isotopes used uh, in vitro or in vivo, 1H we know has a spin of half. The others that have a spin of half are 13C, 15N, 19F, 31P, and 195 platinum, which is very, very rare to be used. So in the given options, only option D has 1H, 15N, and 31P, all three of which have spin of half. So the correct answer is D. You can actually remember the nuclei that have spin of half, since that is the most common that is used. So 1H, 13C, 15N, 19F, 31P, and this. You can just remember the nuclei to answer questions like this. Next question is asking which one of the following is false when considering limitations associated with using 1H as a suitable NMR probe for drug metabolism analysis. So here again, if you look at the drawbacks of 1H, 1H has a low chemical shift range. This is correct and it's a drawback. This will result in spectral overlap. There is peak overlap due to resonance from body fluids. This is also correct because the body fluids are mostly water and that will have a strong 1H signal. So there will be peak overlap. That is, so these two are correct. Decreased JJ coupling is false because 1H has extensive JJ coupling, which results to a lot of multiplicity. So this is false. And the question is asking which is false. So the correct answer is C. Next, the question is asking drug design to enhance bioavailability of a drug involves which of the following steps? Here the answer is, modification of specific functional groups. So suppose we have a lead compound of the drug and we want to enhance bioavailability or other properties. Then we will modify the functional groups. We are not going to repurpose the drug for a different target. Target is fixed. We are not going to modify any probes. Probe is only used to understand the structure of the receptor site. But if we modify specific functional groups, the bioavailability, potency, toxicology, all that can be improved. So the correct answer is B. The next question is asking which one of the following NMR assisted drug discovery methods require labeling of the target biomolecule? So here the answer is NMR solve. If you remember NMR solve, we have to label the target biomolecule or protein to find out the orientation of the receptor site in order to optimize the ligand that binds to the receptor. And we also have to bind the attach a probe to the region between two receptor sites to find out the orientation uh, uh, in order to optimize the ligand, which will, in order to optimize the linker that will connect the ligands between neighboring receptor sites. So NMR solve is the technique that involves labeling the target biomolecule or protein in order to optimize the drugs that will bind to the receptor site and to optimize the linker that will join the different ligands. So the answer is B, that is NMR solve. The next question is asking, ligand mediated chemical shift is the mainstay for which one of the following experiments? So answer is all of the above, because we know from SAR by NMR, basically we are finding ligands for each binding site separately. And the way we are doing this is by ligand mediated chemical shifts. So these are all ligand focused methods where the drug discovery is based on the ligand, and we are optimizing the ligand binding in each site separately, and then we are connecting the two ligands with a linker to get a bidentate ligand that has a much higher binding than each of the ligands separately. And in order to find ligands for each site, the method used is the chemical, how the chemical shift is changing on binding. NMR solve, we just saw in the previous question. Here also, in order to find which ligands are binding, the change in chemical shift is monitored. And ILOE or interligand LOE, these NOEs are used to optimize the linker that will bind two fragments that are attached to two neighboring receptor sites. So here also, these you can see how the chemical shift is changing, that is used for this ILOE. So the correct answer is D, that is all of the above. The next question is asking, which of the following NMR parameters is not useful for determination of protein peptide or protein protein interactions? We know that to determine whether an interaction is happening, variation in peak intensity is important. Variation in chemical shift is also important. So this is important, this is important. Line width variation is important, but that is not in the option. Another is the T2 or relaxation time. So if that varies, that is also important to determine whether these interactions are present. Variance of molecular weight is not used as an NMR parameter. And here the question is not useful. So the correct answer is option D, that is variance in molecular weight. The last question that we will discuss today is asking that for assessment of protein receptor interactions through saturation transfer difference, suppose we are recording the saturation transfer difference spectra of cell plus ligand, of ligand separately and that of cells. So what will saturation transfer double difference experiment give? So this is an experiment. I am discussing this now. So the saturation transfer difference spectra of, so suppose we are not able to separate the protein from the cell. It is a protein that is within the membrane or something. We are not able to separate it from the cell. So in that case, how we study the STD NMR is that we take STD NMR of cell and ligand, and we take STD NMR of only the cell, and then we subtract them to get the STD NMR from the ligand binding. So this is an experiment called saturation transfer double difference, or STDD, because we are taking the difference of two STD NMRs. And these are used mainly for proteins where you are not able to separate it out from the cell, for example, like membrane proteins. So the saturation transfer double difference, you take the STD spectra of cell and ligand, that is X, and you subtract the STD spectra of cells, that is Z. So X minus Z, that is the correct answer, is option B. So that is all for this course. And as always, I'll upload the recordings and you can go through it. And this ends this course. I have covered basic principles of NMR and how NMR is used in determination of structure in nucleic acids, proteins, and in protein ligand protein interactions, and also discuss the basics of drug discovery. So I'll end the class here.